Good morning, everybody, and thank you for joining us today. My name is Josie, and I'm part of the public engagement team here at Alzheimer's Research UK. And I'm delighted to welcome you to the 5th event in our Lab Notes online series for 2022. So I've just got some slides to share with you now as well. So today we will be hearing from Dr. Shahid Zaman and Dr. Stephanie Brown from the Cambridge Intellectual and Developmental Disabilities Research Group. They will be sharing the current knowledge and research into the links between Down syndrome and dementia. But first, before we get started, I'll go over a little bit of housekeeping. So during the event today, you are welcome to switch on the automatic subtitles using the CC button at the bottom of your screen. They're not 100% accurate during the live event, but they will be edited so that they are correct on the event recording. During this event, if you would like to ask a question of our speakers, you need to click the Q&A button at the bottom of your screen, which will bring up the Q&A box where you can type your question. Please do submit your questions throughout the event, and we'll try to answer as many as we can in the Q&A session after the talks. If you are having any technical difficulties, you can use the chat button to let us know, but please use the Q&A button for questions for the speakers. So now we're going to ask you a few poll questions, because while we can't see you due to the online format of the event, we're really keen to know a little bit more about you. So these questions ask about your reasons for attending today, to rate your current knowledge of research, and whether you've attended one of our lab notes sessions before. So the first question is, how would you rate your knowledge of dementia research? Would you rate it as very high, high, average, low, or very low? Which of these best describes your reasons for attending this event today? So do you have dementia? Does a friend or a family member have dementia? Are you a carer? Is your work related to dementia? Or do you not have a personal connection but want to find out more? Or perhaps it might be another reason. And finally, is this your first dementia research event? So have you been to one of our Lab Notes events before? And once we've got a few answers for those, we'll share those results with you back on the screen as well. So I'll just give everyone a couple more seconds to answer those questions for us. And then you should be able to see those on the screen now. So it looks like most people would rate they have an average knowledge of dementia research, and that's great. We hope after today you feel that's improved, and a nice variety of reasons why people have attended, and lots of people it's their first event. So welcome to Lab Notes, and welcome back to everybody that's come along before. So just before we get started and I introduce our speakers, I've got a little bit to talk about our Gifts in Wills. So October is our Gifts in Wills month here at Alzheimer's Research UK. So I want to just share a little bit of information about that. So here at Alzheimer's Research UK, our vision is a world free from the fear, harm and heartbreak of dementia. And particularly during the pandemic, we've seen firsthand how the power of research can really change the future. People choose to support us in many different ways, whether you host a bake sale, run a half marathon, give on a regular basis, or perhaps make a donation in memory of a loved one. Your support makes a real difference to bringing forward the day when we discover a cure for dementia. A third of all of our research projects are being funded by gifts that supporters have left in their wills. This has enabled us to continue our vital research, particularly over the last few years. We know that gifts and wills aren't for everyone, and it's an incredibly personal decision to make. But having a will and making plans for the future is really important to make sure your wishes are recorded and up to date. We have lots of resources that can help you when it comes to planning for the future as well. We've got 3 will writing offers to help you write your will for free in a way that suits you. So with Fair Will and Co-op, you can write your will online or over the phone. And with the Free Wills Network, you can meet with a solicitor. If you have any queries or questions about gifts and wills, please do head to our website, and there should be a link appearing in the chat shortly. So today, our speakers will be talking about the links between Down syndrome and dementia. Once we've heard all the talks, I will return to the screen and we'll move to the Q&A session. So you can submit questions at any point during the event, and we will try to answer as many as we can in the time that we have. Just to say, we can't really provide clinical advice in response to questions about your individual or personal situations. So please do get in touch with our Dementia Research info line or your GP if you do have specific or personal questions. We'll be hearing from Shahid first, followed by Stephanie. But before that, we have a recording to share with you from brother and sister, John and Rebecca. John and Rebecca are siblings who have recently joined a study which is looking into biomarkers of Alzheimer's disease in people with Down syndrome. Shamaya and Jess are research assistants working on this study, and Shamaya is asking the questions in this video. So without further ado, it's my pleasure to share this video with you of John and Rebecca. So I'm going to stop sharing my screen and that should appear. There we go.
Thank you for joining us, John and Rebecca. First, I guess you could just introduce yourselves and tell us a little bit more about what made you want to take part in our research. So John, we'll go to you first.
Who are you?
I'm John, as in my sister here next to me.
Why did you want to do this research study?
Why am I meant to take part in this research? I think my mum asked you lot to ring first, isn't it?
So I think what happened was that somebody told our mum about the study, and then she had a conversation with you about it. And then when you decided that you thought it would be an interesting thing to be a part of, then you spoke to me about it. And we had a little chat, didn't we? And I suppose mainly we thought it would be useful just because it helps other people, doesn't it, ultimately? Not just people with Down syndrome, but also potentially typically developing people who start showing signs of Alzheimer's, and I think we probably all know somebody with Alzheimer's and have seen the effects on the individuals and the people around them. So I suppose for me personally, having obviously the link with John and the link between Alzheimer's and Down syndrome, there's that element, but also it's just I don't know, I think if there's anything you can do to help, we're going to do it, aren't we? That's why I think it'd be a good idea.
Perfect. Do you kind of agree with that, John? Is there anything else you'd want to add, or?
I think Becca said, I think my sister says it all, actually. I like to do the research after I think the first part of the research was very interesting to around the world, that who has or who have not been in this part of this research. It's been wonderful, isn't it?
Do you want to go into a little bit more about what taking part in the research, what you had to do to take part in the research, and which parts you enjoyed then, John? You kind of touched on that a little bit there.
What have you enjoyed so far?
I think the first part of the research is me and Dad into Fulbourn, and it was the first part of the research in Fulbourn is those puzzles and quizzes that with you, Shamaya, and it was really good actually for me to know what the other people who have got those symptoms. But for me, it's so interesting indeed, because we found the blocks and the bits and things around, and I thought it would be a very good research to the others around the world who have around this area and things around. I think it's really good things to find about this research anyway. So it's been very interesting in the first day of the research.
Yeah, so on the first day we did some quizzes and puzzles, so some cognitive tests, didn't we? And we did some ones with blocks and lots of different ones. So you kind of enjoyed that. And can you remember what it was like having the scan on the next day? Did you like that?
I think yes and no. The no part is still all the scanning of things and blurts everywhere inside the brain. And I did choose my certain song that I like to put on the the Spotify, and you can choose what music inside. You can listen to it. You can close your eyes or open your eyes, whatever you do. Just lie down. Don't twitch or anything. You have to stay still for about quarter an hour, like 40 minutes or something inside the scan. So it is good for me because of I like to lay down, have a sleep and things like that. Try not to twitch or anything. That's quite good for me because I'm trying to rest. I like to have a rest.
Yeah, you had to, it can be a bit loud, can't it? So the music helped with that, you were saying, and also that there are some people next door who kind of talk to you and help you through the process and tell you when it's likely to be over and like how far through you are.
Scans and things.
Yeah. So we were do you have anything more to say about taking part, or should I kind of ask Rebecca a little bit about her experience of taking part?
In her or me?
Well, do you have anything more you want to say about it, or shall we see what Rebecca has to say about it a little bit?
I think that's it for me.
So yeah, Becca, you could tell us a bit more about kind of what sort of things the research involved, what things you particularly enjoyed, any bits you found a bit more challenging, and yeah.
So far I've just done one day at Adam Brooks, and it involved I had a CT scan actually separately, one CT scan, and then there was a full day at Adam Brooks, and that involved some bloods being taken, and it involved 2 PET MR scans, one in the morning and one in the afternoon. So I had a cannula in, and the one in the morning, there was they put a little bit of radiation in there, radioactive substance, which is to see the amyloid protein, and then in the afternoon it's a different radioactive substance they put through the cannula, don't they? To see the tau protein, I believe. So that was fun. For me it was quite a nice day out because it was a lot of sitting around, which I don't normally get to do. So I could sit and read my book throughout the day. The scans themselves were, did you tell them that they were so incredibly noisy, weren't they? So that took I suppose that took a little bit of getting used to, because it's quite unpredictable when the sound comes on, isn't it? And there were also some cognitive tests and puzzles. I quite enjoyed them. So yeah, I mean, so far it's been a very positive experience. And then the next in about 18 months' time, it all happens again, but without the for me without the radioactive substance for the scans. So far so good. You didn't like the needles, did you?
At first I don't want to like to have needles in me, but when I get that needle off me it feels much better.
Yeah. So it's only the needle's only in there for a little bit, isn't it? Then it's the cannula, so it's just like a cannula, and then it's not in for that long.
Yeah, we were talking about that, weren't we? So we thought, yes, I would definitely recommend it. From my point of view, you know, I think any kind of way you can inform research to develop a medicine or an intervention that in the future can help other people is a really positive thing to be a part of. We did conclude, didn't we, when we talked about it, that if you really didn't like having injections or something, then maybe you thought maybe you wouldn't recommend it to somebody if they were really scared, didn't you? But yeah, ultimately, I think anything that can inform something to help people in the future is a good thing.
Yeah. Cool. And finally, is there anything else you'd like to talk about or share about your experience at all?
Is there anything else you want to say?
For me, I think not at the moment, but it will be eventually, you know.
You might think of something
I think
in the future.
In the future time. I think for me, it'd be interesting next year, this month, I'll be going back there.
Yeah.
Next time.
Yeah, kind of following it over time and seeing how things change. Yeah.
We both liked getting the photos of our brains, though. That was good, wasn't it?
That's fun.
Yeah, definitely a perk.
That's really interesting, isn't it? Seeing, getting the pictures of our brains.
Thank you both, John and Rebecca, so much for kind of agreeing to talk to us and sharing a bit of your experience. Yeah, and I guess now we'll just stop recording and then we'll just say goodbye, I guess.
Yeah.
Great. I'm sure you will agree that was a really interesting insight into what it's like to take part in research. So I'll now hand over to Dr. Shahid Zaman to tell us a little bit more about the links between Down syndrome and dementia. Over to you, Shahid.
Thank you. I'll share my screen in order to pull up the visuals. Hopefully you can see that.
Yeah, we can see them.
Thank you. So hello, everyone. I'm going to talk about dementia in people with Down syndrome. I will try and explain what is Down syndrome and its importance to dementia, and I will also discuss some of our research findings. But before I do that, I'd like to first present this extract from the BBC News website showing Tommy Jessup, who has Down syndrome. Now, he is a famous actor, but what he was tasked to do was to look into the twice likelihood of a person with learning disability going into hospital and dying compared with the rest of the population. And I think that's an important message to think about, as this population is underserved and there are a lot of health inequalities related to it. Talk about numbers now. I think people are aware there are about 900,000 people in the United Kingdom with Alzheimer's disease or AD. And of those 900,000, there are about roughly 9,000 who have what's called familial autosomal dominant AD, which is it's Alzheimer's disease, but it's caused by faulty genes that are inherited. So if your parent has a copy of this gene, he or she will be guaranteed to get Alzheimer's disease. And you have a chance, a 50% chance of inheriting the abnormal gene. Quite frightening. Out of the whole population, not the population of people with Alzheimer's disease, there's about roughly 41,000 people who have Down syndrome. And so that's about 4.5% of the total AD population. But the majority of them, and that's about 70% over the age of 60, will develop Alzheimer's disease. So the question is, why is there such a high risk of developing Alzheimer's disease in this population? Before we do that, let's look at age. In this graph on the lower axis here, you see age from 30 upward to 90. It shows the age at which you're likely to get a diagnosis of dementia. And it compares people who don't have Down syndrome, that's this axis, compared with people who have Down syndrome. And as you can see, by the age of, let's say, 50, roughly 30 or more % will have a diagnosis. Whereas in the general population, the diagnosis doesn't start to appear and become common until about 70 and 80 and above. So you can see there's a clear shift leftwards in terms of when the diagnosis is made in people with Down syndrome. Now let's look at some history. Here is a picture of August Dieter, who died aged 51 years. Now she had problems for some time with her memory, her ability to communicate because of language problems, and her behavior and mood were also a problem. And it was a puzzle as to what was going on. This chap here, some of you may recognize, is Alois Alzheimer's. He was a psychiatrist at the time who was looking after her, and after her death, he examined some post-mortem brain tissue using the latest techniques and of staining techniques to find out what was going on. The first thing he saw was, here's a cartoon of a normal brain on the left here. This is where approximately where the ears would be. This is the top of the brain, and this is the lower part of the brain. And there's a vertical slice. And if you look at these convolutions, this is the cortex, and compare it to what an Alzheimer's brain looks like. As you can see, there's lots of shrinkage. And if you look at these spaces, which are called ventricles, there's gaps there, which indicates cell death occurring. Here is the hippocampus, which is an important part for memory. And so this general picture is what Alzheimer's would have seen in August Dieter. He would have also seen, using a microscope similar to this, using these silver stains, and this is drawing from his work, what he described as plaques and tangles, which we later found out were made of a protein called amyloid and tau. And here's another picture of the depicting a normal tissue, showing the brain cells, the neurons with the nucleus and the cell body. And here's the abnormal or Alzheimer's version of the tissue, where you see the amyloid plaques, which are outside the neurons, and inside the cells, the neurons, you see the neurofibrillary tangles, which are made of tau. So this is what he described, and the disease was obviously subsequently named after him. So what about Down syndrome? What is Down syndrome? If you look in a person with Down syndrome, if you look at their cells, this is a cell in the nucleus. Inside the nucleus, you'll see chromosomes, and the chromosomes come in pairs. And as you can see, there's 22 pairs, and each of these pairs are obviously identical. They contain the DNA or the genetic information that we all have. And we have another set of chromosomes, the X and Y, and these determine the sex, and this person is a female because she has 2 X's. But she also has an extra chromosome here, which is thus the name trisomy of this 20 chromosome 21. Why is that important? Well, it happens to contain a gene called amyloid precursor protein, APP. And this, we believe, is the cause of the problem. So in summary, genes, especially certainly in Down syndrome, dementia in people with Down syndrome is a genetic form. We believe it's a genetic form of dementia, and it has similarities to the autosomal dominant Alzheimer's or familial Alzheimer's disease, which I described earlier. If so, why is that? So we know, as I said earlier, the APP gene is probably responsible for this. And we know the APP gene is located on chromosome 21. And in people with Down syndrome, they have an extra copy of this chromosome. And that's why it's called trisomy 21. We think that's responsible for the higher rate of dementia. And what sort of abnormalities do you get in the familial autosomal dominant? Well, you get mutations in the same gene, APP, and also mutations or abnormalities in the genes or the proteins that are responsible for breaking down APP when it's translated into protein, called presenilin 1 and presenilin 2. Here's a sort of an example, a little bit further explanation of what I'm talking about. So again, this is the cell body and the nucleus. Inside there's chromosomes. Chromosome 21 will have the APP gene, which is transcribed and translated into a protein. This is the APP protein. And there's the presenilin gene, which is again translated into various proteins, presenilin 1 and 2. And these form this enzyme complex called gamma secretase. And these are kind of the scissors, as it were, which cut APP. And when it cuts it, it forms smaller amyloid fragments, which we think are causing the problems of cell death and also some of the problems which we think are even responsible for what you see under the microscope, the senile plaques or the amyloid plaques. So having an extra gene, as in Down syndrome, causes, we think that's why we get higher rates of dementia in people with Down syndrome, or having a mutation in this gene, which causes problems with breakdown, how well this is processed, or if you have mutations in any of these two genes, causes the autosomal dominant inherited forms of dementia. So the key issues in dementia in people with Down syndrome is that by the age of 40, almost all of them will have the neuropathological features of Alzheimer's. By that, I mean, if you look at the microscope, if someone's died for another reason, they've had a post-mortem examination, you'll see the amyloid plaques and tangles and characteristic features of Alzheimer's. But a person who has these, a living person who has these features, may not have the signs and symptoms of dementia, because the onset of symptoms takes much longer. It takes 10 or 15 years. And that's not just true in people with Downs, it's true with the general population as well, and with people who have autosomal dominant forms of the disease. So there's a very long latency or preclinical phase where there's no apparent clinical signs and symptoms. But there may be changes in the blood and other changes that you don't see unless you look for them. And our research is partly related to that, in trying to understand if we could characterize this part of the disease. So memory and thinking problems. So as I said, these are very common in people with Down syndrome, aged around 40 to 50. And as I said, we've been trying to find out what are the processes underlying this, of using brain scans, as you heard in that very fantastic early, the video you saw earlier, the two participants. And what we found so far is that indeed we do see, you can see these amyloid plaques and tau tangles using the latest technology, using brain scans instead of obviously waiting for a post-mortem. We can inject these traces into the veins, and they go into the brain and they stick to these proteins specifically, show it, and you can see these actual deposits on a PET or MRI scan. So how this is a picture of what actually a pathologist would see if they did a post-mortem on someone with Down syndrome and dementia. In the panel A on the top left, you see the amyloid plaques in the black area there, which is stained with, I mean, it doesn't normally look like this unless you stain it. You see the amyloid deposits in there. And the PET scan works by having a compound called Pittsburgh compound B, which is radioactive and then sticks to the brain tissue in a living person. Then we can take a photograph of that. And same applies to another tracer called AV1451, which would stick to tau, which is shown in that panel D here on the. This is stained for tau, and this is stained for amyloid. So you can see where these compounds are. Here's examples of what we've actually done in people who have Down syndrome who've been scanned. Here's someone with no clinical dementia, and you see these are the eyes, the ears here, and the back of the head. These colors represent a background, so that means there actually is no significant amount of amyloid here. But in someone this is a male, 47, with early dementia, you start seeing hot spots of amyloid here and there. And this one, 65-year-old female with clinical dementia who has, again, lots of hot spots of amyloid. And here's a 39-year-old with minimal binding in this area. If this graph shows you this is showing all people who have Down syndrome that we looked at. On the x-axis, you see the age from 30, 40, 50, 60 upwards. And this on this axis shows you how much amyloid there is in their cortex. And as you can see, as people age between 40 and 45, there's a sharp rise upward to the age of 55 of the amount of amyloid that you can detect in the brain. Obviously, we can do it without having to do a post-mortem, which is great. And then these black dots represent the ones who are more likely to have a clinical diagnosis, and the white open ones who are clinically normal. And that's just reflects the sort of gap you get between the time you get the pathology and the time you get the actual clinical symptoms. Now, if you look at normal, healthy volunteers doing the same test with age here, you see and amyloid here on the left, the y-axis, you start seeing, again, a rise in the amyloid in normal, healthy volunteers, and quite a sharp rise with age. So the same thing is happening in the general population. The difference is in people with Down syndrome, it's happening much earlier. You heard about the study in the video, and this is what we've been doing. This is the ABCDS consortium, which is based in the US, and we are one of the sites. It stands for Alzheimer's Biomarkers Consortium in Down syndrome. We have several visits and people do quizzes and puzzles and have blood sample taken to see if we can detect another way of detecting these changes in the brain. And they have the scans, which were mentioned in the clip earlier. And so in summary, people with Down syndrome have an extra copy of the APP gene, which leads to the overproduction of amyloid, and that leads to deposition. It's amyloid deposition in the brain, which we think causes dementia. Now, in familial autosomal dominant forms of Alzheimer's disease, there's a fault in the APP gene, or there's a faulty gene, the genes that process the breakdown of the APP, the presenilin genes, which we think is the cause of the disease in this population. Now, other forms of Alzheimer's disease are very similar to the above two here, the Down syndrome and the autosomal dominant, especially regarding the formation of plaques and tangles, and many other features too. So studying dementia in this population with Down syndrome, that is, has made many breakthroughs helping us to understand Alzheimer's disease better. Just to finally, I'd like to acknowledge that all the funders, this whole venture is very expensive, and without funding, we wouldn't be able to do any of this research. I'll stop here. Thank you.
Thanks, Shahid. That was really, really interesting. I'd like to welcome Dr. Stephanie Brown to give your talk now, please. Thank you.
Okay, thank you. Hi, everyone. Thank you for attending this Lab Notes talk. I'm Stephanie Brown, and I am an AR UK Research Fellow at the University of Cambridge. So, as Shahid has just mentioned, Down syndrome is a genetic cause of Alzheimer's disease with both a high need and possibility for generalizability. So there are more people with Down syndrome worldwide than other forms of genetically inherited Alzheimer's disease. We know that 70% of deaths in Down syndrome are caused by dementia, compared to 12.5% in the general population. And we know that poor sleep in earlier life increases risk for dementia by 30%. So individuals with Down syndrome are particularly prone to a variety of sleep problems. And of interest in my research are neuropeptides, which are small proteins in the brain, which act as a regulator for certain bodily functions. When we sleep or feel sleepy, this is most likely due to neuropeptides in our brain at work. Similarly, the opposite action of neuropeptides can wake us up and make us feel alert. Findings are suggestive now for a role of sleep-related neuropeptides in the development and severity of dementia. And the role of sleep in dementia is emerging as an important and modifiable risk factor. So orexin is a neuropeptide which controls sleep and also functions to stimulate appetite. If after taking a nap you wake up to find you're very hungry, this is most likely a function of high levels of orexin. We see that in older people with Down syndrome and those who carry the ApoE4 genotype, which increases risk for Alzheimer's disease, orexin levels are lower. All of the orexin producing neurons in the brain are located in the hypothalamus, which is a small region deep within the brain about the size of an almond. The hypothalamus is what we would call a homeostatic regulator for the body, so it keeps balance. It balances wakefulness with sleep, food intake with energy expenditure, and regulates temperature and blood pressure, among other things. It's very perfectly placed to do this, as it receives a very good blood supply from the rest of the body and is well connected to the rest of the brain. So it signals both to the body and other brain areas. In my research, I'm interested in the connection that has been suggested by preliminary research that disturbed orexin signaling may be a factor in Alzheimer's development. When we get impairment in orexin signaling, this causes problems with sleep, which in turn means the positive effects of sleep are lessened and memory is impacted. This then leads to an accelerated dementia process. As orexin-producing neurons are located only in the hypothalamus, we're interested in segmenting this region structurally. Our pilot data show that the size of parts of the hypothalamus are associated with levels of orexin measured in the blood. These specific regions contain the orexin neurons and include our circadian clock, which is our body's way of controlling our metabolism and behavior over the period of 24 hours. For instance, telling us when to sleep and when to wake up. So we're showing here for the first time in humans a direct link between the hypothalamus structure and the neuropeptide orexin, strengthening our evidence that orexin signaling in sleep is impaired in aging Down syndrome. But how might poor sleep be linked to worsening whole brain health? One mechanism is known as the glymphatic system, which, while we sleep, flushes cerebrospinal fluid around the brain to wash away waste products into the blood circulation from where they are safely removed from the body. You can picture this system as functioning a bit like a dishwasher. In dementia, we're beginning to see evidence that this waste removal system of the brain is impaired and doesn't flush toxic proteins like amyloid and tau out of the brain as effectively as it should, which causes accelerated Alzheimer's symptoms. So we're interested in examining the glymphatic system using high-resolution brain images to understand more about it and to help develop treatments that may help it to function better. So I mentioned high resolution imaging. What do we mean when we say this? We'll start with the term Tesla, which was a term named after Nikola Tesla in 1956 to describe magnetic strength. Magnetic resonance imaging, MRI, harnesses magnetic fields to image inside the body. In the clinic, you would expect to have an MRI scan in a 1.5 Tesla scanner. In research, a 3 Tesla scanner is routinely used. And in Cambridge, we have a 7 Tesla MRI scanner approved for clinical use. And this is the only scanner of its type in the UK. This means we have a very strong magnet which improves visualization of the brain. So using 7 Tesla MRI, we can really revolutionize the detail we can see in human brains. We can look at previously difficult to see areas of the brain and delineate their regions with high accuracy, which means we can find parts of the brain responsible for dementia processes. For instance, the thalamus and hippocampus are known areas of early changes in Alzheimer's disease. Using 7 Tesla MRI, we can investigate these areas in great detail. Similarly, the locus coeruleus, labelled here as the LC, is just a few millimeters in size, yet it is vitally important in producing a neurotransmitter called noradrenaline. It communicates closely with the hippocampus to consolidate memory, and degeneration of the LC has recently been identified to be the earliest visible change in the brain prior to the development of Alzheimer's disease. Small areas that surround blood vessels, known as perivascular spaces, are integral to the glymphatic dishwasher system I previously mentioned. And these spaces are difficult to detect in a normal MRI scan, but we can examine them using the advantages of 7 Tesla MRI. So overall, my research uses this high resolution imaging to give us new understanding of how sleep is linked to brain health and dementia. Finally, following on from this project, pathways to patient benefit and impact include examining important links between Alzheimer's pathology and sleep, the therapeutic potential in Down syndrome. This may be through pharmaceutical, medical device, or behavioural interventions. Further to this, future strategies can be implemented for people in the general population with high dementia risk to improve sleep quality or therapeutically target small, important brain structures and orexin metabolism. Thank you, and we'll go back to Josie and Shahid for the Q&A now, I believe.
Great, thank you, Stephanie, and I'll just welcome Shahid back to the screen as well. That was both really interesting talks, so thank you so much for joining us today in Lab Notes, and I'm sure everyone will agree that those are both really fascinating. So just while we wait for some questions to come through the Q&A, so remember you can submit your questions at any time using that Q&A button at the bottom of your screen, but we did have some pre-submitted questions, so I'll just pull those up now. So perhaps, Shahid, maybe this might be one for you, one of the pre-submitted questions. So does an individual know themselves that dementia is developing? And how do we know the difference between sort of normal memory and forgetfulness and dementia?
Yeah, so I mean, it's often difficult for someone to know whether they're having memory. They may have a slight inkling, but what usually happens in clinics, at least, you'll usually get family member, spouse that brings the person to the clinic and explains, my husband, wife is having, becoming forgetful, just being, that's the main usually complaint. But the other things that usually happen, we often think about dementia, Alzheimer's dementia, as being predominantly memory, which is true. But prior to that, you get lots of different other changes, such as mood changes, maybe anxiety, behavioral changes. So those things we do look at very carefully. And remember, I said in my talk that the underlying brain tissue changes occur for many years before you get the clinical diagnosis. And during that period of time, there is likely to be other things that are happening and not necessarily detectable by the person, but you might be able to detect these things in, say, a blood test, or maybe looking at an MRI scan, which may show subtle abnormalities. And a lot of the research is about that, trying to detect that early phase somehow or other, because it's such a long time before you get the clinical symptoms, which means that you're less likely to be able to change the course of the disease if you treat it so late. So that's why it's really important that we define that longer period of time before you get the clinical presentation.
Great, thanks, Shahid. Stephanie, perhaps a question for you. So what impact does the magnet have on the body or the brain in the scans?
So the magnetic field doesn't really have an impact on you physiologically. Maybe some people tend to feel a little bit dizzy when moving in and out of the 7 Tesla, but usually the magnetic field, as long as you have no metal on you, it's perfectly safe and doesn't impact the body at all.
Great, thank you. I know John and Rebecca were talking in the video about it being really noisy and really loud. So I guess that also would be perhaps an impact, but it wouldn't affect you physiologically. Thanks, Stephanie. Perhaps another one back to you, Shahid, following on from that question that I asked you before from Sophia, who asks, what can a patient do after signs in the brain have been noticed before the onset of dementia? So maybe when those first clinical signs have started, perhaps.
Yeah, I think that the main drive is to try and get some help and investigated by a doctor or your health services. Because even though you may get symptoms that might be suggestive of onset of dementia, there are often other things that need to be excluded. A common thing is mood problems, depression. It may be some nutritional problem or some other medical condition which needs to be looked at very carefully to exclude, to make sure it's not that before you go. So any signs, symptoms, I would go and ask. But the biggest risk factor in the non-Downs in general population and the people who are not genetically prone is age. So as you get older and you start getting symptoms that may be suggestive, wanting to be, as a doctor, I would think, there's a higher chance of this person having dementia. But still, it's important to exclude other causes and not be fooled by things that look a bit like dementia.
Great, thank you. I'll ask this question and then perhaps you can both decide who's best suited for it. So it's a question from Rachel, who says, have any studies been done which explore whether there are positive effects of starting people with Down syndrome on medication in the preclinical phase to slow the symptomatic signs of dementia developing?
I can answer that. I mean, there have been trials that unfortunately have failed. One of them was a vitamin E trial. The vitamin E is supposed to have some antioxidant properties, and that was a theory from work in animal work and other stuff. Unfortunately, in clinical trials, it didn't really do any good. But we're all working towards focusing on the amyloid side of things, because, as I explained in my talk, amyloid seems to be still a very important target. And recently, you may have heard in the news, there's been various drugs that very specifically target amyloid by removing it. And the trials have shown in the general population that it's likely to certainly remove the amyloid from the brain and also improve the cognition. And our hope is to do this very soon in our population of people with Down syndrome, is to give the drug before you get the symptoms, a lot earlier, and therefore sort of have a sort of preventive effect of these to some degree. So yes, there are drugs coming online. There have been lots of failures, but there are drugs, and we're very excited about that. And I think our population of Down people will have more. I think my prediction is they're likely to benefit the most.
Great, Thanks, Shahid. Stephanie, perhaps another one for you. So Susie asks, are sleep problems in themselves indicators of the onset of dementia?
Yeah, so this is one of the quite interesting things about sleep, is that if a lot of the time sleep problems can also be a symptom of dementia, especially things like middle of the night wandering, for example. We think this is playing into some issues that I mentioned with the circadian clock. We don't really know why sleep gets so disturbed in Alzheimer's. So while you're healthy, it's a risk if you have poor sleep in earlier life. And then as you kind of progress into a dementia status, you would then see exacerbation of sleep problems. Yeah, so it can be an indicator. Yeah.
Great, thank you. Shahid, did you want to add something there?
To add something there. So in people with Down syndrome, we know probably nothing to do with dementia. They tend to have a lot more cases of obstructive sleep apnea. So they stop breathing at night for various reasons. And so if you correct that, people who may present with problems with thinking and focusing and behavior, that improves. But like Stephanie said, it's certainly a risk factor for diagnosis. So if you have a sleep problem prior to being diagnosed, it's probably a contributing factor to having a greater chance of having a diagnosis.
Thank you both. We did a lab notes session last year, actually, on the links between sleep and dementia, whether it's a symptom or a risk factor. So if you wanted to hear more about that, you could have a look on our website, which we'll pop a link in the chat later on to find that. We've got another question from Alison. Again, I'm going to ask it and either of you can jump on it. So is gene editing ever likely to be possible to remove the amyloid producing part of the gene?
It's a great question, and there are people, at least in working in Down syndrome, trying to attempt something of silencing, not necessarily using CRISPR, but silencing the APP gene. And why do I feel more quite sure that APP is in Down syndrome, the extra copy is responsible? Because there have been published cases of people with Down syndrome who had no diagnosis of dementia into their 70s, and when they died, they had postmortem. It was found that they indeed did have all the extra genetic material in homes and 21, except for that bit where there wasn't an extra copy of APP. So from that, we can conclude that the genetics is pretty fundamental, because everyone else gets the neuropathological changes, but these particular cases, when they didn't have the extra APP, didn't get the disease, which is amazing.
Great, thanks, Shahid. I've got one more question that was a pre-submitted question. So is there anything that you recommend to a young adult with Down syndrome that they should avoid or begin over or above the normal healthy lifestyle to reduce or delay their risk of dementia?
Yeah, I mean, I think the general point here is that whatever applies to the general population applies to people with, in terms of dementia, Alzheimer's dementia, to the people with Down syndrome. I know there's only a few studies to demonstrate this, but certainly I would, clinically, I would recommend doing the same things as recommended for the general population: cardiovascular health, diabetes, controlling all lipids. Having said that, people with Down syndrome tend not to have hypertension, they tend to have low blood pressure, and they have other risk factors that are not as common in the general population, such as thickening of the arteries from lipids or fats. So there might be slight differences, but I think it doesn't do any harm to do all those things anyway. That includes, socializing, learning new things, keeping the brain active, and even dental care as well. So all those things, and hearing. So all those things are just as important in people with Down syndrome.
Thanks, thank you. Perhaps I'll just ask you both one more question. So this is from me. So Stephanie, what are you sort of most excited about for your research project, and what's ahead, and what most excites you about the future of dementia research?
Oh, that's a good one. I think one of the most exciting things based on my project at the moment is that obviously there is a lot of research into sleep at the moment, but there's not too much in Down syndrome, and it might be a very good way of looking at sleep, because we pretty much know that they are going to develop Alzheimer's disease, so we can look at it earlier. And I think that's a really good opportunity. And also the role of neuropeptides is going to be really interesting, I think.
Great, thank you. And Shahid, the same question for you. I know you've already mentioned sort of amyloid and stuff, but anything else that sort of really excites you about the future of dementia research?
I think if you look at the history of Down syndrome, and I alluded to that a little bit, I didn't go into detail, but so many times looking at Down syndrome has helped us to understand in the general population Alzheimer's disease. And I think we're aiming for prevention here, and the amyloid hypothesis there, which was described, is the one that the whole industry has been working on, trying to target amyloid and to reduce it. And the thought is that causes the problem at the start. And I think this is the most exciting period for them. You saw in the chat that lecanemab has been approved by the FDA, the American the drugs regulators, and I think that's where I'm really excited. Trying it out in people, and if it works, it will really tell us whether we're on the right track in terms of modifying the disease, even in the general population. The problem with the general population is, of course, that not only do they have the amyloid and the tau, but with age, because they tend to be older than the people with Down syndrome when they get the diagnosis, they have other complications and other neuropathological complications, such as Lewy bodies and all sorts of other vascular problems, which may complicate the likelihood of success. So I think that is about prevention, and the clinical trials I'm really excited. We're getting there.
Amazing. Thanks so much both. And just as we approach 12 o'clock, I think we'll end it there. So thank you so much both for joining us. That was both really interesting presentations and a really great Q&A as well. So I'll say goodbye to you both, and thank you.
Thank you.
And I'd also like to thank John and Rebecca for sharing their experiences of taking part with research with us. And a massive thank you to Shamaya for recording their discussion as well. And just before you go, we've got a few poll questions that we'd love you to answer again, just to give us a little quick insight into how you found today. So first question, was the content of the talks, did you think it was too technical, about right, or too simple? Would you recommend lab notes to your friends and family? And have you learned something new today about dementia research? So again, I'll just give a couple of seconds for you to answer those. So we will be sending out a more detailed feedback survey to you soon as well. And then we'll pop those answers on the screen. So fab, most people thought that the talks were just about right, and everybody would recommend, and everybody's learned something new. Perfect. So as I said, we'll be sending out a detailed feedback survey, and also the video recording will likely to be next week once we've sort of edited the subtitles and got that ready for you. Please do complete the survey, as we'd appreciate any feedback you can give us about the event. And then I've got a couple more slides to share with you as well. So I know there may have been some questions that we weren't able to answer today, and somewhere you might need some more personalised information or guidance on where to turn. Our Dementia Research info line is there to help. They can answer questions and also signpost to other sources of information and support. So please do get in touch with them. You may also be interested in finding out more about our Join Dementia Research service, which can match you to clinical research studies. This is open to anyone over the age of 18, and you do not need a dementia diagnosis to take part in studies. You can find out more and sign up to Join Dementia Research on their website. There's the link in the chat for that as well. And if you've missed any previous Lab Notes events, you can watch them back in your own time, as they are all available on the Lab Notes webpage or on our YouTube channel. There's the sleep and dementia symptom or risk factor talk, as I mentioned there, just at the bottom. So the link is in the chat for that too. And finally, I just want to say a huge thank you for joining us today. I hope you found it interesting and useful, and we hope to see you again soon.